Hello, today we are discussing somatic embryogenesis. So let us first see what is somatic embryogenesis. Somatic embryogenesis is a process by which the somatic cells, that is the body cells, they developed into differentiated embryos, and from these embryos a new plant is regenerated. So here the somatic cells or tissues, even the haploid cells can be developed into embryos. So this was first induced in carrot suspension culture by Stewart et al. And Reinhardt also produced embryo from callus in carrot through suspension culture. So when we define somatic embryogenesis, it is the production of embryos from body cells. So by taking just a piece of plant tissue, from that we can produce embryos from that piece of plant tissue. Okay, that is called embryogenesis or somatic embryogenesis. So coming to the types of embryos, we know that there are two types of embryos. Zygotic embryos, this embryo is developed from fertilized egg or zygote. So this is the normal embryo, where by the fusion of male and female gamete we can produce embryo. Now non-zygotic embryos are those that are developed from somatic cells. These somatic cells, they develop into embryos, and they are called adventive embryos. Then if they are formed from unfertilized eggs, then it is called parthenocarpic embryo. If the embryo developed from male gametophyte, then it is called androgenic embryo. So types of embryos are zygotic embryo and non-zygotic embryo. Zygotic embryo is the normal embryo that is formed from fertilized egg, and non-zygotic embryos are of three types. Somatic embryo, which is developed from the sporophytic or the plant body cells. They are produced in vitro condition. Then parthenocarpic embryos and androgenic embryos. So this is how we produce a somatic embryo. Here we can see that they have isolated a single cell, and this from single cell a group of cell is produced by cell division or callus formation, and in this cells their differentiation is initiated and then from this differentiated cell embryo is developed. So we can see the globular stage of embryo, the heart shaped of stage of embryo, then the torpedo stage of embryo, and from the torpedo stage a plantlet is formed. Okay, develops into a normal plantlet. So this is how a somatic how they produce somatic embryo. Now when we come to the importance of somatic embryogenesis, we can see that the higher propagation, it gives higher propagation rate. Okay, it can more number of individuals can be produced from by using somatic embryogenesis. Then suspension culture, through suspension culture also we can produce somatic embryos. Then it is used for artificial seed production and production of somatic embryos is a labor saving process. Now coming to the stages of somatic embryogenesis, there are three stages. First stage is callus initiation. From the plant tissue, callus is initiated, and then embryo development and maturation. So the callus is differentiated so that it develops into the embryo. Then finally plantlet formation occurs. So this is a diagrammatic representation where they have shown how from a single cell that single cell develops into a embryo. So we can see the different stages of the embryo: the globular stage, the heart shaped stage, then the torpedo stage, and then it develops into the plantlet. So from the torpedo stage a plantlet is developed. So this is a diagrammatic representation. Now let us see what are the factors affecting somatic embryogenesis. First factor is character of the explant. So when we can see that young or juvenile explant, or when we isolate tissue from young plants, it produces more somatic embryos than older plants. And also according to the plant tissue, the complexity of the plant tissue, it produces embryonic cells in different frequencies. So from the same mother plant we can see that the embryonic callus is produced at different frequency. So coming to the characters of explant, so the explant should be from a young a plant, and the cells also matter. From where the cell is taken, that also matters. Then the next thing is growth regulators. We can see auxins and cytokinins play a key role in the development of somatic embryos. Auxin in combination with cytokinin, it induces the embryogenesis and also the growth of the embryo. So the concentration should be specific, that is effective concentration ranges are 0.5 to 27.6 microns, and for 2,4-D it is 0.5 to 10.7 micron level. And the cytokinins are also used in the primary medium, and the concentration is 0.5 to 50. And cytokinins also, they fasten the somatic embryo maturations, especially the cotyledon development. The cytokinin helps in fastening the cotyledon development. So these are the two growth regulators. One is auxin, the other one is cytokinin. So both these are used in combination with each other. Next is abscisic acid. Here in abscisic acid, it is used in inhibitory levels, that is it helps for it promotes the development and maturation of somatic embryo. At the same time, it inhibits the abnormal proliferation and the initiation of accessory embryos. Then nitrogen sources are also important. Nitrogen sources also are added to the medium, and then high potassium levels and low dissolved oxygen levels also, they prevent somatic embryo regeneration. So growth factors, along with growth factors, nitrogen source and potassium and oxygen levels are also very important for the development of somatic embryos. Now the medium for somatic embryo is normal MS medium, which is supplemented with growth factors, and the temperature for the medium is 25 degrees Celsius, and 16 hours of light is provided by fluorescent bulbs. The intensity of light is 16.75 millimolar per meter square per second square, and this light and temperature is provided for the period of 8 weeks. The callus induction frequency and regeneration frequency can be calculated for optimal somatic embryogenesis. Now let us discuss the type of somatic embryogenesis. There are two types of somatic embryogenesis. One is direct somatic embryogenesis, the other is indirect somatic embryogenesis. Direct somatic embryogenesis means here the embryos are initiated directly from the explant without the formation of callus. Here the callus formation is absent, whereas in indirect embryogenesis the callus develops from the explant, and then in the later stage embryos are developed. Okay, so we can see how the picture of direct and indirect embryogenesis. This is the picture of direct embryogenesis. Here we can see that the first picture shows the explant, and in the second picture we can see that there is no callus formation and direct plant are produced. In the fourth picture we can see that the root initiation also started. Okay, but in indirect embryogenesis there is callus formation. Here we can see that indirect somatic embryogenesis picture is given. Here in the third third picture we can see that there is the callus formation, and from the callus these callus cells are differentiated so that it develops into embryo, and from each callus cell we can see that multiple embryos are growing. This is a research paper, a journal of rice research, where they have induced embryonic callus cells, and a whole plant is regenerated. Here they have given the concentration of hormones they have used. Now coming to the advantages of somatic embryogenesis, we can see that this method can be used in controlled culture condition, and then a lack of material is not a limiting factor. That is, even with one plant we can develop many embryos. Here high propagation rate can be seen, then germplasm conservation is possible. Similarly, it is a labor saving process, and also we can get as a result we can get disease free and virus free plants. So and there are many advantages. One is if minimal culture conditions are given, and then even if we have only one plant, we can develop many embryos out of it. Then high propagation rate is occurs, and then germplasm conservation is possible. Now coming to the disadvantages, only few species they respond to somatic embryogenesis, and also we can see that somatic embryogenesis shows very poor germination rate. Right, and then we can also we have seen that the cultured cells are very unstable. So and that is a major limitation for commercial exploitation. Okay, so only few species respond to somatic embryogenesis. The next thing is very poor germination rate of the embryos is seen, and the third one is the embryos are unstable, and therefore commercial exploitation is not possible. So I hope you understood this section. So if you have any doubts, be free to ask for any clarifications. Thank you very much.